Hello, my name is Lise Maloney. I'm the Director of Business Development for Sciessent. Thank you very much for attending this presentation. My talk today is on what you need to know when selecting an antimicrobial additive for your medical devices. So first we'll go over some factors that are driving the use of antimicrobial additives in the market today. And we'll go over some strategies for choosing the best technology for your specific application. And then we'll shift gears a little bit and take a look at some recent antibiotic resistant testing that we've conducted on different medical grade polymers. And lastly, we'll take that a little further with a case study looking at central venous catheter applications. So this is just a brief introduction to Sciessent. We are the successor in interest to Aegon Technologies as the result of a management buyout 4 years ago. We're headquartered in Wakefield, Massachusetts, just north of Boston, and we have agents in Europe, Asia, and Latin America. Our flagship technologies are the Aegon antimicrobial additives, which consist of silver, copper, and zinc formulations for use in both plastics and in coatings. We service the healthcare, industrial, and consumer markets with these technologies. And we also have some additional technologies for high-performance textile finishes to address odor management and durable water repellency. And so now we'll take a look at some factors that are driving the use of antimicrobial additives in the healthcare space. The number one driver is healthcare-associated infections, or HAIs as they're known. The problem was really identified back in 2002, and data was started to be collected and published in 2007, where they looked at sort of the baseline data, what's going on, the magnitude of the problem. And CDC and the Department of Health and Human Services set out a goal to completely eliminate healthcare-associated infections. And these are infections that you get if, for example, if you're going in for a knee replacement, and while you're there you end up with a surgical site infection. That's considered a healthcare-associated infection. And so with this goal of eliminating these infections because they feel they are preventable, CMS, the Center for Medicare and Medicaid, started to implement some reimbursement restrictions. So for certain healthcare-associated infections, healthcare facilities are no longer reimbursed for these infections. So there's financial incentive as well as outcomes incentives to really try and minimize these infections. So just in March of 2014, the CDC put out a progress report to see how we're doing since we first looked at the baseline data and since they've been implementing these strategies to help reduce and eliminate these infections. And what we see now is that about 1 out of 25 patients will develop at least one HAI during their stay in the hospital. The original data was 1 out of 20, so we are seeing progress in this area. This results in about 75,000 deaths per year, and previously we were at about 99,000 deaths per year. So again, some progress is being made. If we look at some of the specific types of healthcare-associated infections, overall we see a reduction in things like central line-associated bloodstream infections, surgical site infections, MRSA bloodstream infections, and C. difficile infections. But we are seeing a slight increase in catheter-associated urinary tract infections. So ultimately, more is definitely needed. We are nowhere near that goal of completely eliminating these infections, and in some cases, we aren't even making some progress. So if we take a little further look into why aren't we reaching that goal of eliminating these infections, what we see is that compliance to these HAI guidelines that are out there is surprisingly low. Data from CDC shows that less than 40% of U.S. hospitals are in full compliance with the guidelines to help eliminate HAIs. And I think a lot of the reason for that has to do with the training and resources that are required to implement these best practices are definitely added strains on these healthcare systems, which are already limited. And so it takes a physician that's been doing something a certain way for 20 years now has to change his practice, and it's something that's going to take more time and more training to fully implement these guidelines and best practices. So what we see is healthcare facilities are really turning to their medical device manufacturers to help them come up with innovative ways to further lower these rates of infections and to supplement their infection prevention programs. And embedded antimicrobial additives is certainly a great strategy for helping to further reduce these rates of infections. And it's something that can be implemented as sort of a second line of defense. It doesn't require someone changing their practices to implement these. The second factor that's driving the use, and in my opinion is really going to be the greatest factor moving forward, is the problem of antibiotic resistance. The rapid spread and development of antibiotic-resistant organisms is really an alarming concern. CDC put out this threat report in 2013 to really try and bring attention to this developing problem. There's a statement in that report that says that if we don't act now to try and prevent further antibiotic resistance and to try and limit the spread of the already existing antibiotic-resistant organisms, we'll soon be in a post-antibiotic era. So that means that we'll have simple infections, a cut on your hand, and people will be dying from these infections because we'll no longer have antibiotics that can treat these infections if we don't start to limit the use of the existing antibiotics that we have. So in terms of strategies to help address this problem, on the use of antibiotics, there's a lot of trying to have a more prudent use of the antibiotics that are being delivered. And the second component is really how do we limit exposure and limit risk of our patients in the hospital to being infected or exposed to these antibiotic-resistant organisms. And this is where I think antimicrobial additives can play a really big role in potentially limiting that risk and exposure of patients to these antibiotic-resistant organisms. The antimicrobials must be effective against antibiotic-resistant organisms in order to have an impact in this area. But assuming that they do, I think they can play a big role in how healthcare facilities are addressing this problem of antibiotic resistance. And just to go over a few of the statistics from this report, 2 million people become infected each year in the US with resistant bacteria. 23,000 of those people will die from these infections. And it is purely caused by the overuse and the misuse of antibiotics. So if someone goes to their doctor for a cold and says they want an antibiotic, if that cold is caused by a virus, that's a misuse of that antibiotic. And by exposing that patient to it, you're now exposing the bacteria within their system to potentially be able to adapt and develop resistance. The statistics in there are extremely alarming. More than half of the 90 million prescriptions that are written annually in the US are unnecessary or inappropriate. So really half of our antibiotic use is not appropriate and necessary. And just to expand on that a little bit, this is a timeline from that report, and you can see I'll use methicillin as an example. And it's developed, the antibiotic was first introduced, it's on the far left-hand side of your screen, in 1960. And by 1962, just 2 years later, they've identified the first strain of methicillin-resistant Staph aureus. So you think of how long it takes a drug manufacturer to develop a new antibiotic, and it just takes 2 years for a bacteria to develop resistance to this strain. The future pipeline for antibiotics is pretty limited. Drug companies see this type of data and realize that it's not a good opportunity for them. They're looking at developing more chronic disease drugs because the amount of resistance and how quickly these bacteria can adapt is just surprisingly fast compared to the dollars and time it takes to develop new antibiotics. And another factor that's driving the market, so not only do we have the healthcare facilities obviously very aware of changes in reimbursement and the problems with antibiotic resistance, but now we have consumers really hearing and learning about these superbugs, these antibiotic resistant organisms. And they're not only being discussed in infectious disease conferences and microbiology conferences, they're making headlines in the mainstream media, and this is just a sampling from the past 6 months. And so potential patients, consumers are educated and realize the concerns with these superbugs. And so when they go into a facility for treatment, they want to make sure that their exposure to these antibiotic-resistant organisms are limited. And these are not sensationalized headlines. They are really depicting the problem with antibiotic resistance, and it is a huge problem that needs to really be addressed right now. To take that even further, consumers now have choices that they've never had before. Medicare.gov created this hospital compare website where if you're going in for an elective knee surgery, for example, you now have the ability to access this website and put in 3 hospitals that you might choose to have your procedure performed at, and you can see how they rank in terms of quality of care. You can look at their infection rates specifically. So now consumers have knowledge and information that they've never had previously, and they can make informed decisions on where they want to go for their care. So this is a big driver as well. Healthcare facilities now have pressure from consumers as well as from government agencies and insurers as well. So now we'll take a look at some strategies you can use when choosing a technology for your antimicrobial medical device. There are a lot of technologies out there, and this is a very simplified view. I won't get into the chemistries and the differences among them. What I will try to do is provide some strategies to help guide you towards what's best for your application. I wish it was a one-size-fits-all area or technology, but unfortunately it's not. It really is dependent on that end-use environment and really what your goals are for your devices. So a technology that may be great for one application may be completely inappropriate for another, just based on what the requirements are and how it will be used and where it will be used in the body. So with that said, before anyone dives into a technology search, which is always kind of where we usually want to start, I highly recommend that you first take the time to really define your product requirements and understand the parameters that are going to be important for determining if this antimicrobial is appropriate for your use or not. And really, once you've defined these, then allow that to guide your search, and it will get you ultimately to the best technology that's out there for your application. So just a few of these that are really critical for the antimicrobial component is, again, that end-use environment. Is this a device, is this a patient monitoring device that's going to sit bedside, that will be cleaned between patient use, or is it an indwelling device that will see high flow, constant flow, that you need efficacy out for 30 days, a year potentially? Is this a reprocessed scope that will have to see 100 plus reprocessing steps? So understanding what that end-use environment looks like is very critical, and will again help you decide which technologies may be appropriate and which ones are not. Understanding what target organisms are clinically relevant to your device. For several of them, it's pretty well documented. Central venous catheters as an example, it's pretty well known which organisms are important for that device and which cause device-related infections. You want to understand that, and then again make sure that the antimicrobial technologies you're evaluating are effective at killing those target organisms. Antimicrobial performance: if there is currently a device on the market in your space that has already established a certain performance level, you want to make sure that you know what that is and that the technologies you're evaluating can meet or exceed those performance levels. Otherwise, there's some sort of unwritten rules with FDA that 3 log, 4 log are probably where you want to be. Minimum duration of efficacy. So looking at the useful life of the device and how long you want the antimicrobial effectiveness to last. Those are very important. If the device is used for less than 24 hours, disposable, fairly easier duration to handle for efficacy. Again, if this is a permanently implanted device or a long-term indwelling device, is there a certain period of time where the risk of infection is higher? In the case of a permanent implant, potentially while the surgical site is healing, there might be a higher risk of bacteria infiltration and infection, and that might be the duration that you're looking for efficacy. Another important consideration is having a good understanding of your current manufacturing processes and capabilities on site. You want to sort of ideally know how you would want to add this antimicrobial. Can it be compounded directly into the polymer so it's sort of seamlessly introduced into your current manufacturing process, or would you like to look at a surface treatment with a coating application? If that's the case, do you currently coat your device? If you don't, that's going to be adding another manufacturing step, and potentially have to look at doing that with a contract manufacturer or bring those capabilities on site. So I think it's really good to sort of understand the manufacturing capabilities and what potentially could be utilized to add the antimicrobial. And lastly, you want to make sure that the antimicrobial supplier has experience with medical device applications. You really want to use them as your partner, as an extended team member for the product development process. You are the experts in designing the medical devices, but your supplier should bring that expertise and knowledge of the antimicrobial and how it can potentially interact and how it should be evaluated for ultimately regulatory approval of the device. It's really, we've seen a lot of companies go about it alone and then ultimately sometimes come back and ask for help. I can't stress that enough. I think the most efficient and straightforward way for getting through the product development process and regulatory approval is to really partner with that antimicrobial supplier. And this is just an example. We've actually created a formal program called MedMap, Medical Device Market Acceleration Program, to help our medical device customers. And we can be inserted anywhere along this pathway, right at the concept, looking at regulatory strategies, helping develop claims that might be the easiest to get through the FDA process. We can help in terms of test plan development, test method development. You think of simulated use or duration testing, we can help with some of that method development. And then on the regulatory side, we can be a part of reviewing 510Ks, helping draft the language, certainly in terms of supporting data for those filings. It's getting a medical device through that doesn't have an antimicrobial is a not an easy process on its own, but now when you add an antimicrobial technology that potentially isn't an area of expertise within your company, it's really helpful to be able to have a partner that has experience with this area and can certainly provide some guidance. So to look a little further on the regulatory side, in 2007, CDC put out this draft guidance, FDA put out this draft guidance for 510Ks for antimicrobial medical devices. The guidance document is still in draft form today, and I assume that it's going to stay that way, but it really is the only formal source of guidance that the industry has to go on. So I would recommend that the R&D engineers and the regulatory team become very familiar with this and the requirements within it. And these are just really some specifics from that draft guidance document, specifically related to the antimicrobial and information that you will need to provide in that 510K. And again, this is areas where hopefully your antimicrobial additive supplier can help you address each of these. The most important one is the indications for use relative to the antimicrobial component. So what types of claims do you want to make? This really should be set early on in the process because it should drive your verification and validation testing. So if your marketing team has the idea of wanting to mention infection at all, infection prevention, treating infections, that all automatically requires a clinical trial. So typically you want to have very limited focused claims that you can prove within your verification and validation testing. Next thing is the comparison of the device to a predicate device. This is always necessary when you take the 510K route. And typically, there isn't an exact device that's your same device with the same antimicrobial already in it. So typically what you'll need to do is have multiple predicate devices. And usually it can be your existing device would be the predicate for the device itself, and then you would use a predicate device for the technology. And this can be, hopefully it's something that's a higher risk device. If you're doing a permanent implant, it wouldn't be acceptable to have a predicate device that was maybe a patient monitoring device that's not indwelling in the body. So understanding what you can use as that predicate for the technology and which devices are already approved using that technology is really critical to identify an appropriate predicate device. You'll need a detailed description of the antimicrobial chemistry and any ancillary components. So the ancillary components might be things if you're applying the antimicrobial in a surface coating, you'll have to provide the chemistry for that surface coating as well. The mechanism of action and spectrum of activity of the antimicrobial agent. So this, again, your antimicrobial additive supplier should be able to help you with this. How does the technology that you're using kill bacteria, and what bacteria or organisms does it kill? And that should be identified through those target organisms that you were looking, that you had identified prior to searching for your technology. What's the release mechanism and release kinetics of the technology? This, you really have to characterize how it releases and then what that release profile looks like over time, over the duration of use. You'll have to identify the concentration of the antimicrobial within the device and also characterize the minimum effective concentration. And usually our recommendation for this minimum effective concentration is setting your lower limit for your spec for the concentration at that minimum effective concentration and doing all of your verification and validation testing at that minimum spec. And lastly, the distribution kinetics, toxicity, metabolites, and degradation products of the antimicrobial additive. You need to have identified them and understand what's going to happen to this technology once it's introduced into the body, and that's from a safety standpoint. And typically for most technologies, there is a body of literature that can be accessed. Sometimes you'll have to hire a toxicologist as a consultant who might be able to write a risk assessment in terms of the addition of the antimicrobial agent. So these are some of the things that, beyond the traditional 510K, these are specific to the antimicrobial that absolutely need to be in that 510K. And so now we'll shift gears a little bit, and I want to present some antibiotic resistant testing data that we've just recently completed. So going back to that concern of antibiotic resistance, we wanted to make sure that our Aegeon technologies are effective against the antibiotic resistant organisms that are out there. So what we did was we looked at 6 different medical grade polymers. We incorporated our technologies into them, and then we tested them against 4 known antibiotic resistant organisms. The materials that we looked at are ABS, polycarbonate, polypropylene, PEEK, silicone, and polyurethane. We figured we wanted to get sort of a broad spectrum look at different medical grade materials that could potentially use an antimicrobial additive in them. And the methods we used are ASTM standard antimicrobial methods. For most of the materials, we looked at both 4-hour and 24-hour contact times. For the silicone and polyurethane, we were testing existing catheters that are on the market, so we looked at the 24-hour time point for that, thinking that we want longer time durations for those. The organisms that we tested, coming right from that CDC report on antibiotic-resistant organisms, they are the top concerns for antibiotic-resistant strains that are out there. The biggest one right now that not as many people are aware of is CRE, carbapenem-resistant Enterobacteriaceae. And this is a group of organisms that certain strains within it are are resistant to every single antibiotic that we have out there. So that's why its hazard level by CDC has been put in the urgent category. That basically means that, yeah, for some of these strains, it's sort of a 50-50 shot. If the patient can clear the infection themselves, they can recover, but if not, there's really no antibiotics that can be used to treat these infections. Methicillin-resistant Staph aureus is probably one of the most known, and infections with MRSA are caused both in the community and also in the healthcare setting. There are some antibiotics that are still effective against that right now, so this is in that serious category as determined by the CDC. Vancomycin-resistant enterococcus, or VRE. This again is in that serious category. A lot of infections, but there are some antibiotics right now that are effective against it. And the last organism is multi-drug resistant Acinetobacter. Again, some antibiotics still effective against it, but several that aren't. And so how I broke up the data was by material. So if we first look at ABS here, what we did was we had a control that we compared it to after the contact time of either 4 hours or 24 hours. So we're inoculating the surfaces of these treated and untreated polymers, and then recovering any viable bacteria that we can after 4 hours and 24 hours. So we've calculated log reduction on the left-hand side of the slide. I've also included the percent reduction. Some people, that's a little bit easier to understand. So what we're seeing, if we look at CRE, that urgent level, really concerning organism that's out there right now, the technology is very highly effective against CRE. After 4-hour contact time, we're reducing more than 5 log of the bacteria on the surface, and that equates to over a 99.99% percent reduction in bacteria on the surface. And after 24-hour contact time, we're at almost a 7 log, and essentially we are not recovering any viable organisms. We're at the limit of detection of the assay, and we're also limited by what grows on the untreated surface for that comparison. But we're not detecting any viable bacteria within 24 hours against CRE. If we look at MRSA, very similar data. At the 4-hour contact time, a 6 log reduction, and slightly better after 24 hours, almost a 7 log reduction. VRE, we're at almost a 5 log reduction at that 4-hour contact time and over a 6 log reduction at 24 hours. And for Acinetobacter, over a 6 log reduction at 4 hours and almost a 7 log reduction at 24 hours. So the technology in ABS at this concentration that we've chosen to test is very effective against all 4 of these organisms, and we were very excited. We had not tested CRE in our lab, so this was sort of our first exposure to it, but we are very effective against non-resistant strains of Enterobacteriaceae, so we were hoping to see similar results, and we're very happy when we did see that. Now we'll move on to polycarbonate. And again, if we look at CRE, we see highly effective, over a 6 log reduction at 4 hours and almost a 7 log at that 24-hour time point where we're not recovering viable bacteria. If we see MRSA and VRE, these are gram-positive bacteria, and traditionally gram-positive bacteria are a little harder to kill with silver. And so at those shorter time points that we're looking at, we do see lower rates of reduction, but still significant, almost a 4 log reduction against MRSA at 4 hours. Once it's in contact for 24 hours, we are seeing over a 5 log reduction. And these were not optimized loadings. We really just tried to take sort of a nominal loading and take a look. So there's room for improvement if shorter contact times are desired for these types of material. And VRE, we have about a 2 log reduction at 4 hours, and then it jumps up to over a 6 log reduction at 24 hours. And again, Acinetobacter, which is a gram-negative, highly effective at both the short-term 4-hour contact time and the long-term 24-hour contact time. So now if we move on to polypropylene, again, we see very similar results. Those gram negatives on the end, the CRE and the Acinetobacter, highly effective at both the 4-hour and 24-hour contact times. If we look at the gram positives, MRSA and VRE, again, we see that it's some kill, starting to kill at that 4-hour contact time, but takes a longer duration of time to see very highly effective levels of kill, greater than 5 log and greater than 6 log for both organisms. But overall, in polypropylene, the technologies are effective. And now we take a look at PEEK, which is an engineered polymer that we see use in implantable devices. And we can see here that with all 4 organisms, we're highly effective at both those short contact times and the longer times as well, over a 5 log reduction across the board. So now we'll take a look at the silicone. And this, as I had said before, the previous samples were injection molded coupons that we tested in the lab. These are actually approved central venous catheters that are on the market. So it's tubing. And again, we tested it for that 24-hour time period and wanted to see how well it does against CRE. These are organisms that we've traditionally never tested. They haven't really been around except for in the last few years. And again, gram-negative bacteria, these silver antimicrobials, are highly effective against them. We recovered no viable bacteria, no CRE bacteria after 24-hour contact time. MRSA over a 3 log reduction, VRE over a 6 log reduction, and the Acinetobacter over a 5 log reduction. And these are polyurethane catheters, and again, we see very similar results. Gram-negative bacteria, very effective. Gram-positive, 5 log reduction, I would consider that very effective as well, and VRE almost a 4 log reduction. So we were happy at the end of this, our thought is we need to start addressing antibiotic resistance, and we wanted to make sure that our technologies are effective against it. And we were very happy with the data and pleased to see that our technologies can be used to address this problem of antibiotic resistance. And so now we'll just take a little further look into central venous catheters, which are those polyurethane catheters that we just showed on the last slide. So this is testing that had been done previously, not looking at resistant strains, but Staph aureus. This is the susceptible strain of Staph aureus. And you can see we wanted to test out to 45 days of simulated use to make sure that in an application like that, we want to look at long-term efficacy and not just a 24-hour type of exposure. And again, Staph aureus is that gram-positive, so out at 45 days, we're starting to see a drop-off in efficacy, but highly effective out to 30 days. E. coli and Klebsiella are gram-negative and very effective across the board, even out to that 45 days. So the Klebsiella pneumoniae is a susceptible strain, but the one we tested in the resistant, the CRE strain that we tested in the antibiotic resistance was a Klebsiella as well. So what we like to see with this data is that we're equally effective against resistant strains as we are susceptible strains. To the silver, it really doesn't even recognize that it's a resistant strain because the antibiotics have such a different mechanism of action from the silver. So, again, another opportunity for these embedded antimicrobials that have very different mechanisms of action from antibiotics. Yes? No, that's a good question. These are all directly compounded into the polymers and extruded with them. Thank you for that clarification. So these are not coatings. The technologies can be applied within coatings, but in all of these cases, we compounded them into the polymers, so they were on the surface and throughout. So these polyurethane catheters were studied in 2012 in a clinical trial for their use in preterm infants, which is an area for antimicrobials that really hasn't been studied a lot. Usually it's adult populations. But this investigator knew that there was a very high rate of infection in these umbilical venous catheters, which are basically used to access the bloodstream right after birth. And so these neonates, 22% rate of infection in control catheters. That's an extremely high rate of infection. With the use of the Aegon-treated polyurethane catheters, they saw a 2% rate of infection, so a 90% reduction in the rate of infection. And this data has actually led to a recommendation in the SHEA guidelines that were published this past summer for the use of the Aegon antimicrobial technology in central venous catheters. So again, in this application, the technology is directly compounded into the polymer. So it's throughout, it's in the lumen and on the external surface, and there's no concerns of coating durability or degradation over time. And so just to summarize, the rapid development of antibiotic resistance, the continued need to reduce HAIs, and the ability of medical devices to act as carriers for these pathogens is really what's driving the use of antimicrobials in the healthcare sector. And before deciding on a technology and starting your technology search, take the time to define the product requirements up front and understand the regulatory requirements and let those really define or guide your search for finding the technology that's best for your application. And lastly, and I think most importantly, that when you do find a technology, make sure that that supplier is well versed in FDA regulated products and that they can support you and really use them as a part of your extended team. I think it's the best way to efficiently and successfully get through the product development and regulatory cycles. So thank you for your time, and if you have any questions, I'd be happy to answer them. Yeah, so we've, most of our partners will do the mechanical testing with our technology, but the amounts that we are going into the polymers with, we haven't seen mechanical problems. And even on some of those catheters, that one used in the neonates, it's a very small multi-lumen catheter. So obviously there is concern where you could start to have some impact on the mechanical properties, but it typically takes very high loadings. The loadings we go into, our partners haven't seen problems with mechanical properties, but I'm sure there is a point you could get to where you would start to impact that.
Sorry, I missed the beginning of your presentation. Excuse me. Hello. My question is this technology silver-based? I mean, your name says it all, I guess, AG-ion, is that right?
Yes, so as Sciessent, we have silver-based, we have copper-based, and also zinc, and we have combinations of all three.
Okay.
But primarily on the indwelling medical devices, we typically use our silver-only technologies. We have started to look at some of the silver-copper combinations, but yes, all metal ion based and primarily silver.
Okay, thank you.
Yep. All right, thank you very much.